Welcome to the NCT Data Science Seminar. This seminar is jointly hosted by Heidelberg AI. A warm welcome to Florian Markowetz, our speaker today, and to Lena Meyer-Hein, the chair of the Data Science Seminar Series. She will host this event. Lena, the stage is yours.
Yeah, good morning, everyone. It's my great pleasure to welcome Florian. Florian is actually professor of computational oncology at the University of Cambridge and senior group leader at the Cancer Research UK Cambridge Institute. He's also a co-founder and director at Taylor Bio, a genomics startup. He holds a degree in mathematics and philosophy from the University of Heidelberg and also a doctorate in computational biology from Free University Berlin. So he's originally from Germany. He received several awards, including the Royal Society Wolfson Research Merit Award and the CRUK Future Leader in Cancer Research Prize. I personally first met Florian, as far as I know, at an EMBL event where we started speaking specifically about AI failures. And then I got really excited, actually, when he told me about his new paper, from which he actually thought I was a reviewer, but I wasn't, which is called All Models Are Wrong and Yours Are Useless, Making Clinical Prediction Models Impactful for Patients. And yeah, I'm very much looking forward to talking also the behind the scenes information on how nature handled the phrasings of his work and all that. So yeah, stage is yours, Florian. Welcome.
Thank you very much. Can you hear and see me? I hope you can. Good. Thank you very much for the kind introductions and the invitation to speak in the seminar series. Heidelberg is very close to my heart. I come from a little town called Bensheim, which is 20 minutes to the north of Heidelberg. I studied in Heidelberg. My scientific career started on the 8th floor of the main building of DKFZ, which many of you might have never seen because you're now in all these fancy newly built buildings that just were sprouting everywhere in the last 20 years. So I talk to you today about my paper, All Models Are Wrong and Yours Are Useless. It came out in Precision Oncology. It went through a couple of rounds of reviews like papers do. And as soon as the academic reviewers were satisfied with the content, the professional editor actually read it. And she wrote me an email and she said, well, we accepted your paper, but there's something about the tone. It's not all positive. Could you maybe just completely rewrite your paper to make it sound more cheerful? And I wrote a very polite no back. But as a response, I have started to include a warning slide in my talk. There will be strong views and harsh truths. And if you need to sit down, if you feel a little bit unstable, sit down now. Make sure you're under supervision if you feel a bit weak today. What I will talk about are clinical prediction models, and I have a very broad view of what clinical prediction models are. This is a slide I took from my friends at the Mark Foundation Institute for Integrative Medicine in Cambridge. What you see on the left, and I actually wonder if I can, oh, that's not what I wanted. If I can make you see my pen, turn on the laser pointer. This is good. Here's a laser pointer. Look at this. What you see on the left, these are patients. The little circles are supposed to be biopsies or some measurements. And from that, data is derived. And the way I think about this for this talk, it doesn't really matter what type of data it is. Could be clinical data, radiogenomics, metabolomics, tumor genomics, digital pathology, anything goes. But there is a problem, there's a field out there of people generating clinical prediction tools, and it takes some or all of these data, somehow massage them with some AI, statistics, machine learning, and then hopefully tell the doctors how better to treat the patient. So that's the promise of clinical prediction tools, is to help treat patients. You can identify these papers as not just being algorithmic papers by the words they use. So the papers I talk about generally have phrases like, this model could be used in the clinic. Now, we're all scientists, so nobody says this model could be used in the clinic. We're much more careful than that. What people actually say is this model could probably have the potential to maybe be used in the clinic. So scientists do this because they're careful and because they want to lose all accountability for what they are doing. Because imagine you write a paper with a new model and it is not used in the clinic. Somebody could come and say, hey, you wasted all this taxpayer money we funded you with. And you say, no, you didn't. I never promised it would be used in the clinic. I just said it could probably have the potential. And you're safe and you can apply for the next grant. The other key phrase that people use, or the key term when we about the type of models we talk about, is precision medicine. Precision medicine is a completely badly defined term, which somehow implies that medicine as it is not very precise. But if we had better biomarkers, better computational models, we could somehow tailor these treatments better to patients. And that is a good thing. So you will see in the papers that I talk about, you identify them by phrases like, our model implements the promise of precision medicine, or some bollocks like that. Now, this means, from what I just said, I'm talking about a particular type of science here. Science, the way I think about it, comes in two flavors. There is, on the left, there's discovery science. This is defined by whether or not it generates insights. So you work in a model organism like, I don't know, Drosophila, and you're interested in a particular transcription factor, and then maybe your experiments really show how this transcription factor is important in the development of the fly embryo. So this is an important insight. At no point are you saying you help a person with that. You're fine. You're in discovery science. I will not insult you during this talk. But maybe you're over here. You're in translation. The translational science means actually we take stuff that comes from more discovery science, basic science, algorithms, data, ideas they have generated, and we use them on clinically important problems to translate the basic science to the clinic. And translational science should be judged by actions. Does this actually lead to some actionable change in the world that made people's lives better? So this is, for me, the difference between discovery science and translational science. One is interested in insights, the other one is interested in actions. I'm having trouble with the controls today. So how are we doing with clinical prediction tools? Well, I'm showing my age with the type of memes I included. So this is from a movie called Spider-Man. This is Spider-Man's girlfriend. And she looks at him and she says, tell me the truth. I'm ready to hear it. So what does Spider-Man say? Well, Peter Parker, your new clinical prediction model. Many similar models already exist, and she's still holding it together. She's still holding it together. And then he continues, and none of them are used, and she starts crying. So there's a whole bunch of biostatisticians who do meta-analyses and analyses of clinical prediction tools. This is a guy called Martin van Smeden. Another person is Gary Collins from Oxford. And they make these collections of the landscape of clinical prediction models. And I stole this slide from one of Gary Collins' talks. So you see here an overview of review papers of how many of these clinical prediction models that try to help people are out there. And look at the top left. There's 408 models of chronic obstructive pulmonary disease prognosis. 408 models. I mean, just think about it. There was a person out there who said, oh, look, there's only 407 models so far. I should be the one to make it now to 408. I mean, what's wrong with these people? And then when all these models get reviewed, the summary of this paper by Ballou 2019 is that actually none of them are being used in practice. Most of them are not being validated. They usually are just trained on very small data sets. They're pretty much useless. So in a way, this is not just 408 models. These are 408 useless models for COPD. None of them is helping anybody. And you will be very hard-pressed to find any used models anywhere in this whole list of hundreds of models. So I think it's not unfair to say that 99% of computational models never get used for patient benefit. And that might be an underestimate. It might just be 99.999999% of models that never get used. It is really an exception. If you meet somebody who says, you know what, we have a paper on a model and we are actually using it in the clinic now. Almost never happens. So that made me think about when is a prediction model successful? And there are different standards for success. The academic standards are very clearly defined. You need an algorithm that is novel. If you predict something with a Cox proportional hazards model from the 1970s, I mean, just go away. I mean, who needs this, right? It has to be a neural network. The other clear indicator that you're successful is the number of citations. If you have been cited by many people, even just in passing in a drive-by citation, was an important paper, an important contribution. And if your paper is a bit new and it hasn't been cited very often, well, at least you can rely on the glamour factor of the journal and you say, look, I'm in the New England Journal, which has an impact factor of, I don't know what it has, 57, I made the number up, but really high. And all of these things may mean I'm successful in my job in helping patients. The problem is, I think these are all clown reasons. None of them actually show that you had any impact beyond your own CV. What actually makes a prediction model successful is that it actually does help patients out there because, as we discussed at the beginning, we're in translational science and the goal is to have real-world actions. So what real success is, it must make people's lives better and not just yours. So it must have an impact beyond your CV. Ideally, what you generated, the model, is something that can be routinely used. So it's not just a one-off in an academic lab. I mean, it's pretty nice that you have a friend who's a doctor and that doctor said, well, I try out your model. This is good. It's not bad. I'm not saying anything negative about it, but it's not routine use. And ideally, routine use means that you don't do it in the center of excellence. I mean, the DKFZ is special. The place I'm here is special. So that we can do stuff others can't. Okay, that's a given. But if you want real impact, you just have to look bigger. So when I do the talk here, I talk about making it work in Peterborough. Peterborough is a small town, 45 minutes from Cambridge, which we look down upon. It's a bit rural. So I wonder what it would be in Heidelberg. It has to work in Neckargmünd, okay? That's where it has to work. So it has to work somewhere outside the centers of excellence, and that is actually real success. So given all of this, I challenge you. I believe all the models you have generated are useless, as are mine. And in the questions, you can try to change my mind if you want to. Now, having fun about other people is good, but I give you some examples of my own papers, and I will explain to you why they are failures when it comes to having real-world actions. And I start in 2012 with this paper in Nature. It's called the Metabrick paper. If you work in cancer genomics, you know this data set. It's a huge 2,000 patient breast cancer study. It has been cited 5,000 times. The data have been downloaded 3,000 times. It's a major academic success in a very good journal. What we did in this paper is we looked at breast cancers and we defined 10 classes, 10 different types of breast cancer based on their copy number and gene expression profiles. And then we looked at survival data and said, look, these 10 classes matter because some of them do very well and some of them don't. So these computationally defined classes really reflect differences in the disease. Bingo. Now, here's the question. Are these 10 subtypes used anywhere in the world to routinely diagnose breast cancer patients? Nope. Are these 10 subtypes used in Cambridge, where they were invented? Or in Vancouver, where they also were invented? Are they used in the places where they were invented to diagnose breast cancer patients into 10 different subtypes? Nope. Are there any plans in Cambridge to maybe tomorrow use these 10 subtypes on their patients? Nope. And that is for paper that is 12, 13 years old. So we had a lot of time. So what is the problem? Well, there's at least two problems with this paper. The first one is, I told you part of the analysis of the subtypes is based on gene expression data. Have you tried to get routine gene expression data in the clinic? Yeah? Haha, doesn't exist. It's an academic subtype. Academic researchers love gene expression. Single cell nowadays, spatial. You never get it routinely in the clinic. So if you work on gene expression, you already know you will not have an impact in the clinic, at least not directly, because the data set you're working on is not routinely accessible. The second problem with this paper is, well, we got 10 subtypes. What do you tell a patient now? Oh, look, Mrs. Smith, you're in the reddish subtypes. You'll do well. Oh, sorry, Mrs. Brown, you're in the brown shitty subtype. I guess you'll do badly. That's it. There's a difference. That's what we can tell them. What we cannot tell them is what action is best suited to treat the cancer of this subtype. So this misses an important piece of the puzzle, which is to say, well, you're in subtype 6, which means we now do the following. So it has to change the treatment in somehow. And as presented here, and also with the research of the last 12 years, we're not at a point where these subtypes guide treatment decisions. And that makes the whole paper, no matter how academically impactful it has been, translationally useless. Why do people do subtype studies? Sorry, can't hear you in the back. Because machine learners love unsupervised methods. The good thing about unsupervised methods is there is no ground truth. So you will always come up with some clustering, and you just need to make a story about it. So machine learners love clustering, which is why people like me do it a lot. So we followed up on this paper with another paper in colon cancer. Again, very nice journal. This time we clustered gene expression data from colon cancers, finding 3 subtypes. But we were a bit smarter now. So first we noticed we were not the only ones who had subtypes of colon cancer. There was a worldwide consortium that had it. So we had another paper in a nice journal, just making consensus subtypes, having like now a view from all over the world on how this disease should be stratified. And then because we knew gene expression was a bad data type, we spent time on using immunohistochemistry to use a routinely useful data type to predict the gene expression subtypes. I sometimes call this talk how to lose collaboration partners and alienate my funders, because one of the authors of this paper I was good friends with no longer talking to me because I asked him, I said, hey, are these subtypes now being used in the clinic? He's like, of course not. And what do you mean, of course? He's like, yeah, well, don't think we ever meant to use them in the clinic, which I don't think is what we promised when we talked in the papers where we talked about precision medicine. So this is another example here where we have subtypes of a disease which could be predictive of something and which somehow should have an impact on patients, and they're not used on anything except maybe cell lines. The reason here is, again, a lack of an action. If I tell you you have a colon cancer of the serrated subtype, what do we do with this? We don't know what to do with this information. So finally, my third example is from just a couple of years ago. I'm getting better and better. I'm getting closer to being useful. This is again in breast cancer. And this is now a multi-omics machine learning predictor of breast cancer therapy response. Aha, therapy is in action. So this is the type of paper that should be useful because it looks at data at the diagnosis of breast cancer and then predicts whether chemotherapy will work or not. This is very useful information. And if you had a patient where chemotherapy is just not working, you would not poison them with it. Can be very, very useful. There's a little bit of a problem here with gene expression, but that's technical. Maybe we can overcome it. This paper, is this used now? It's 3 years old. Is this now used worldwide to guide treatment decisions? No, of course not. Is it used in Cambridge to guide treatment decisions? Nah. Are there any plans to use it tomorrow in Cambridge to guide treatment decisions? I don't think so. And with this paper, I'm much more surprised than with the others, because this is one of the closest things I've worked on that actually has a clear use in the clinic. And for me, it shows how badly developed the paths between academia and the hospital are, that there are no systematic ways, at least in Cambridge, to translate these kinds of models into the clinic unless you have lots of friends there already and you kind of use your buddy network. But I don't think that's a sustainable work to do science. Now, this was all a bit negative. The last thing I want to tell you is, if you ever read papers in these top journals, I mean, I selected this. These were Nature Medicine papers. It's just, the papers I write. And you're very impressed by them. I give you a question you should ask the authors. It's like your burning sword of truth to cut through the bollocks. It's a very short question. Memorize it, note it down. The question is, so what? Hey, I have new subtypes of disease X. So what? Hey, I have a multi-omics predictor of blah. So what? So what means, how do you use this? I mean, how is this helping anybody? And you will find that most researchers have no good answers to the so what question, and that makes me very sad. Now, that was very negative. Let me tell you a couple of more positive stories and see why they are more positive. So what you see here is a thing called a capsule sponge. This was developed by my colleague, Professor Rebecca Fitzgerald in Cambridge, to identify people at risk of esophageal cancer, cancer in the food pipe. If you have reflux, anybody here, reflux, Gaviscon, you eat Gaviscon, you're at high risk of cancer of the food pipe, esophageal cancer. It's the acid from your stomach coming up, changing the tissue in your esophagus. And the question is, how do you find these changes indicative of cancer somewhere down here, close to the stomach? Well, this is where this little device comes in. It's a little pill on a string. You swallow the pill. In your stomach, it expands into a small sponge. And then within a second or two, the sponge is pulled out through your esophagus and collects cells. So this is a cell collection device. Then a pathologist looks at these cells and sees whether some of them look weird. The problem this device had to becoming widely used is that there were not enough pathologists to do this analysis. So we came in and we said, oh, look, we use deep learning. This was some years ago. We used convolutional neural networks to analyze the images of the cells that came off the sponge to do the job for the pathologists. And it's a triage-driven approach, which means we don't do the full job of the pathologist, but we pick up the easy cases. We reduce their workload by 60% or more even, and the hard cases they can spend more time on. And the reason this is now being used actually worldwide, they're now introducing this device in the US, is because one of my students decided to become a CEO of a company. And there is a company called Cited now, which does all the kind of things academics don't think about, mostly as answering the so what question, doing all the steps to take an academic success and a nice paper and having a real product that helps people. My second example is my lab's current flagship paper, which is on chromosomal instability. Chromosomal instability means that the tumor's genome is very, very messed up. And these cancers like ovarian, esophageal, pancreatic, some lung, they're very hard to treat because they have very few recurrent events. So you don't find biomarkers. It's just a huge mess, a chaos. So what we did is we used some pretty standard machine learning, some mixture models, some non-negative matrix factorization, to define copy number signatures. These are patterns in these genomes which are indicative of what went wrong in the cell to mess up the genome in such a way. And we show that these signatures, they're called CX1 to 17, that they are predictive of platinum response. So if CX3 is higher than CX2, you're predictive sensitive, and if it's the other way around, it's predictive resistant. So we have a paper where we have a clear medical need. We have something that can be computed on shallow whole genome sequencing, which is clinically achievable. It's DNA, not RNA. And we have some evidence that it can guide treatment decisions. Is this being used worldwide in the clinic? No. Is it used in Cambridge? No. Are there plans to use it tomorrow? Yes. Oh, I'm sorry, I really struggle with the controls here today. And the reason we have plans is that, again, some of our postdocs started a company and started to answer all the so what questions. And trying to figure out what is the best way to use this. I mean, we have an academic research area too, where we do clinical trials with those, and we try to get it into the clinic. But the main driver is actually having somebody outside of academia very clearly define what the answer to so what is. What is the real impact of that technology? Where can it help the most? Now, these two examples where both involve startup companies. And I don't want to leave you with the impression that the only way to have impact in the world is having a startup company. So my third example is not from me. It's from my colleague, Paul Farrow, who you see down here. And depending on where I am on your screen, I might be standing in front of him, in which case this is him. This is a tool called Predict Breast, which is hosted by the NHS. That's the National Health Service in the UK. It is a Cox proportional hazards model to predict whether adjuvant chemotherapy would work, yes or no. It uses data which every clinical GP, like general practitioner in the UK has. Is this ductal carcinoma in situ? What was the age? Menopausal? What's ER status? This is not full-grown gene expression data. This is not spatial single-cell transcriptomics. This is data every doctor has with a very established statistical model. And this is a web page they spent a long time designing to make it useful. And this is now being used by tens of thousands of people every month from all over the world, doctors, patients, to make the right decisions for their breast cancer. Now, there's no company involved here. There's no money involved here. And it turns out the story of how they got to this, what I think is the most successful clinical prediction tool I know, is it was a long and hard uphill battle. They didn't have the papers in the glamour journals because, remember, if you use a model like Cox proportional hazards, nobody likes you. They had no major grants. My own funder, Cancer Research UK, turned them down several times, thinking this might not have any impact. They had to rely on small philanthropy. And it is, if you listen to the story, it's a really long uphill battle to get this off the ground and working. And everybody just did this in their spare time or on the side or money that was just left over from other grants. And I think it really shows the disconnect we have in academia between what is actually useful and what we value in our grants and papers. And for me, that really very clearly shows this gap. So when I talk to my friends at CRUK headquarters, I tell them the story and I say, you need to change the funding criteria for your grants. You have to put much more emphasis on actual implementation and being useful and not just novelty. A Cox proportional hazard model might be much better than a neural network. So people look at this and there's different directions. I think academia is burning. Other people think this is just fine. So what is the mindset of somebody who looks at this and thinks this is fine? Well, some people look at this and say, Florian, you're a bit too ambitious. The bar is too high. You see, it will just take more time. So there's no success immediately. I mean, drug development takes 20 years. I mean, so to have a medical prediction tool be useful, you just can't do this from today to tomorrow. I mean, it just takes a long time and then things will be fine. The problem I have with this is not that it takes time. It will take time. The argument is completely valid. It will take a long time. But that most people who use this argument, when you ask them, well, what are you doing during this time? What are your plans to actually make it useful? They have no answer. So if it takes time is only an excuse for not doing anything, then I'm just not very happy with this answer. So let us delve into the moral shortcomings and character flaws of people who think that it takes time is a good answer. So the first problem is what I call ivory tower syndrome. And I have a quote I love from a guy called Duncan Watts, who became famous some years ago with network theory, 6 degrees of separation, like the game you play with Kevin Spacey and the Pope, where you list friends. Anyhow, forget about it if you don't know what I'm talking about. So he has this quote, which I will read to you. For 20 years, I thought my job was, as a basic scientist, to publish papers and throw them over the wall for someone else to apply. I now realize there is no one on the other side of the wall, just a huge pile of papers we have all thrown over. So I call this ivory tower syndrome because there's the idea, I'm a scientist. My job is done when the paper is published. Everything else somebody else should do. And this is true. I mean, my examples with the startup companies show you that at some point this needs to leave academia. But the problem with this attitude is people stop with their responsibilities too early. They think the paper is the last thing and then I forget about it. But there's many steps you as a scientist have to do just to find somebody who might be interested in your work and might actually implement it, even if you don't want to do it yourself. And so many of us just don't do the work in identifying them. If I look through my window down here, I see the headquarters of AstraZeneca. For much of the stuff that I do, they would be a prime example of a prime candidate of somebody who could implement this. A new drug I develop, a new biomarker. But if I just sit here, publish my papers, and hope that somehow they see my papers and get excited, we'll never get anywhere. So you actually have to look over the wall and you have to very proactively engage with people outside of academia to make any impact in the world. The second problem is herd syndrome. We live in a world where implementation and practical impact are not very valued, which means they are not really described in papers, which means if you are new to science and you read papers, and you write your own papers, you just say, well, I don't talk about real world impact or implementation because nobody else is doing it. I just do what the others do. I just adhere to the standards in my field and all the kind of stuff about helping patients. That's just not what we do, which I think is very sad for a field like translational science, which is defined by helping patients. Now, you're at DKFZ, you're like me here in Cambridge. You're leaders. You know what leaders do? They're not sheeple. They don't just sit there in the middle of the herd. Leaders lead. So that means you, as leaders, have to sit down and think, hey, is this actually the right way we're doing things at the moment? Okay, nobody is talking about implementations in their papers, but maybe I, the next leader, maybe I should. And you change the way you write your papers. The third problem I see is that people understand all of this, but they think they're snowflakes and that their projects are snowflakes. And they say, well, nothing I've ever done in my life had any patient impact, but I feel it in my heart. This time will be different. This project is so special to me. I just manifest patient impact by thinking very hard about it. And so you have all the best intentions, but without a plan, it will lead nowhere. So that is my 3 reasons why people just say it will take time and still nothing happens in the end. Because they think it's other person's job, because they think I'm just stay part of the herd and not do anything special, or they just have no plan and just hope for the best. Now, what can we actually do? Now, I have something for you that you can do in your next paper to make the world a better place. So if you write a translational paper that somehow talks about patients and precision medicine and all this stuff, your discussion section, which is the most boring section in any paper, because people just reiterate what they already said 3 times. Your discussion section should contain an implementation plan. And by that I mean, you need to answer questions like, look, the model we just presented, this is actually addressing a key clinical decision. This is the decision we are addressing with this. So it could potentially be useful. There is an action attached to it. We can use it with data that are routinely collected. We have thought about what evidence we need to generate in the future to convince our clinician friends. And we actually have thought about this thing called medical device regulations, which is what you have to do with something to get it into the real world. Now, I'm not saying that you have to have solved all of these things already, but I think by the time you write your paper and you argue that you have made some important contribution, you should at least have a line of sight to how it can be used. And you should have some ideas and should have thought about the obstacles on your way until it can get used widely. And you should have an opinion on how best to overcome them. This might be hard if you're a first-year PhD student, but if enough people push for this, even the old fart PIs might change the way they write their papers and think about these questions at some point. And obviously, once the paper is out, don't just sit on it, actually do some of these steps. The problem with this is, currently there are no incentives for implementation. So if you want an academic career, there's nothing, I mean, this sounds good what I just said, but you need to remember I am a fully tenured, very well-funded senior researcher and professor in Cambridge. It's in a way easy for me to say this. It will be much harder for you to do something about it because your career still depends on doing novel stuff like neural networks and publishing in Nature and all of these things. And it will be a very slow change to change the incentive structure of science to reflect more usefulness. But until we do this, the systemic bias that we have against implementation harms patients. There is so many money and hours of your time, my time, spent on useless stuff if we don't have a very clear line of sight to how we help patients. And this money might just be spent better somehow else. The problem is bigger than just you and me. So there's many different types of people and institutions that have to all work together to make the world a better place. You and me, we are the researchers. We just need to implement more. We have to think more about what comes after a paper. The journals, they need to publish this kind of stuff. I mean, I'm very sad seeing journals like Science Translational Medicine, which have the word translational in the title, just publishing the same run-of-the-mill papers that Nature Medicine would also publish. It's mostly what I consider basic research, and the key part of translation, which is implementation, is still undervalued, even in journals that have the word in the title. So journals, if you're a journal editor somewhere there, change your ways. Now we come to hospitals. Hospitals are a problem because hospitals are generally underfunded. And I don't know if you've ever talked to a medical doctor. They are difficult people, very conservative, do no harm, blah, blah, blah. So getting hospitals to actually innovate and change their ways, share data more easily is very, very hard. Here in Cambridge, there's first steps now. They finally started to organize their own data in the hospital. They started to kind of combine different data sources. So there's first steps, but this will be a big problem. The funders, they just need to fund you for actually implementing stuff rather than to come up with the 409th useless model in your field. And universities, senior leaders in departments, they just need to promote you based on these merits rather than just publishing useless papers. So with that, thank you very much. Let me sum up where we are in this argument. We started by observing that 99% of models never get used for patient benefit. I taught you a mighty sword of a question, the so what question, to challenge authors of fancy papers to explain how the heck they think this is helping anybody. I taught you how to think about the it takes time answer and to overcome your own biases, the ivory tower syndrome, the herd syndrome, and the snowflake syndrome. And we discussed that the first step you can do is to add an implementation plan to your next paper. And we discussed that this would be a good step to overcome the systemic bias that we currently have against implementation, and that you're a small part of a bigger change that has to happen, which includes all these other people. With that, thank you very much. You can read my paper in Precision Oncology. I got lots of feedback from people. When I have a strong opinion, I usually send it to lots of people because I want it to be strong but not stupid. The feedback I got was usually positive from the computational people, which I found surprising because there's this whole methods matter crowd out there, and I thought I'd piss them off by saying that maybe your model doesn't matter. But they were like, yep, that makes sense to us. The only negative comment so far I got is from the doctors. One of my friends on this list, just called my paper naive and short-sighted and thinks I'm completely and utterly wrong. And I was just very surprised by that. So let's see what you think of it. And I'm happy to discuss this in the question session now. Thank you very much.
Thank you so much, Florian. That was very interesting and entertaining.